Good afternoon, ladies and gentlemen. Welcome to this afternoon's presentation. This talk by Dr. Eleanor Jones from Fera Science Limited has been pre-recorded. Afterwards, we will have Professor Nicola Spence and Dr. Belinda Phillipson from DEFRA join us via Zoom to answer any questions you may have.
Hello, my name's Dr. Eleanor Jones and I work for Fera Science Limited. Thank you very much for the invitation to speak today, and apologies for not being able to make it in person. I was going to talk today about the analysis on genetics that we've done for Asian hornets. We've been doing this analysis since 2016, but I was going to update primarily about what happened last year in 2023. So by way of background, the Asian hornet Vespa velutina is a hornet species that's found across much of Southeast Asia, from which it has expanded in an invasive way into South Korea in the early 2000s, from whence it's jumped to Japan in 2012, and it's also invaded Western Europe, starting at around 2005. These invasion events are thought to have started from fairly low numbers of founders, but even so, you would consider them to be highly successful. They've spread extensively from their first point of introduction. So if we look at the expansion into Europe in a bit more detail, it was first found in 2005, and it has expanded rapidly since then into the Benelux region, Germany. It's gone down into the Iberian Peninsula and also crossed into the Italian Peninsula as well. This map is taken from an EU Life project website, and it hasn't been updated particularly intensively. It was last updated in 2019, and the dark red shows places where it's been found in 2018 and 2019, and in paler red it's places where it was found previous to that. And as we know, we had the first finding in the UK in 2016, and so the presence is also noted on this map in the paler colours, and then where the more recent findings were, it's in the darker colours. So the region of interest is the UK mainland, and we had our first incursion, our first arrival of an Asian hornet in 2016. And this photo just at the bottom is that first hornet that was found, recovered and sent to Fera for analysis, and we nicknamed it Harriet. She's a female worker, and I think she was from Tetbury. So every year since that first year, there have been about 1 to 3 nests found annually, and they're shown as the orange squares on this map, and they're sort of found not quite at random. They're certainly found more to the south of the country than the north, and we also have single hornet findings. So these are just hornets that have been found on their own. Sometimes they're found next to things where they've probably been imported on, for example camping equipment or bits of machinery brought in from abroad, and sometimes it's not entirely clear where they've come from. And these seem to occur perhaps even more haphazardly across the UK. So we know that these hornets are arriving, and we know that they can successfully breed for 1 year before we exterminate the nest. The pattern was very much broken in 2023. Where instead of having 1 to 2 nests, we found 72 nests at 56 locations. These weren't all laid by individual queens. We know that 14 of them were primary and secondary nest pairs. So 14 of those were the primary nest, and then 14 were the secondary nest that the same queen had gone on to form. But still, it's a remarkable uptick in the number of nests that we found. The big map shows the full extent of where the nests were found, and there's quite a concentration down in the southeast corner of Britain. So what happens with the Asian hornet individuals and the Asian hornet nests that are found and destroyed? So the efforts that go into reporting, finding and destroying the nests are shared across a great number of people, including members of the general public, beekeepers, APHA bee health inspectors, and the inspectors will find and destroy the nests, and they will then package them up, ideally refrigerated, and they get couriered to Fera for analysis. Also, any individual hornet that's been caught in a trap or has been caught as an incidental, maybe hand netted or has been found by someone and reported, those are also sent in tubes of ethanol to Fera. And what do we do with them? Well, the nests are dissected, and then there's genetic analysis is undertaken on a range of the individuals from the nest and on all the lone hornets. So when the samples arrive at Fera, Fera is a very large scientific site with many different teams, but the team that coordinates everything is the NBU National Reference Laboratory, and they coordinate the processing of the samples. So they receipt all of the samples in, they dissect the nest, and they take subsamples of individuals from the nests, and they also receipt in the lone hornets, and they'll take a sample, so a leg from the lone hornets, and they send the samples on to the molecular teams for genetic analysis, but they'll also send some of the samples over to entomology for further dissections to try and identify things about the hornets. And there's just a photo of two labs here. I'll just see if I have my pointer. This is the NBU lab where the samples are receipted in. There's a fume hood at the back, which is used for dealing with any nest that has been covered in pesticide safely so that it doesn't cause any harm to people. And then this is one of the molecular laboratories. I've taken a photo through the window, and it has a big banner that says Plant Health Quarantine Facility, because much of the work that we deliver is actually for plant health as well as for bee health. So with the morphological identification of the individuals, what we want to do is we want to pick out the queens of the nest, and we also want to pick out the females that have been laid and provisioned such that they're going to turn into the next generation of queens, so the gynes. And within the National Reference Laboratory, all of the adult male individuals are sexed, so the males are separated from the females. Just use my pointer, but I think it's very obvious. So the females have the sting and the males don't have the sting. And then all of the females are subsequently weighed. And everything that's above 0.6 grams is considered to be a potential gyne or queen. And this is a number taken from one of the papers out there. So all the females are weighed, everything above 0.6 grams is considered to be a potential gyne and is passed on to entomology. The entomologists take further measurements on those suspect gynes. One of them is the maximum mesoscutal width, where they're looking for a threshold of 4.5 millimeters across the tegulae, across the adult head. And anything that's above 4.5 is considered to be a potential gyne, and everything under that is more likely to be a worker. This photo just illustrates the potential size differences you can be looking at, that this is a queen from a nest and this is a worker from the same nest. Those ones that have already been identified as being potentially gynes on the basis of weight are also dissected. We haven't the time to dissect everything. They're dissected by the entomologists who are looking for the presence of fat bodies, because any gyne is going to have to overwinter, so she needs a certain amount of fat to help her get through hibernation, whereas the workers typically don't carry fat or don't carry much fat. And they're also dissecting to look for the presence of eggs, and these eggs would be produced by the queen that's the queen of that nest, so the one that's out there laying all the eggs. And this rather revolting picture here just illustrates that, where you have the lots of little eggs and then you have the far larger eggs that are presumably making their way through to be laid. Dissections are undertaken by the National Reference Laboratory within Fera, and they dissect the nests, but partly to get an idea of how large the nest is. So the size of the nest is recorded, and characteristics like the number of combs is also recorded. And they also take out the adults that they're going to identify to being males and females, and they take samples of each life stage within the nest. So they take everything from the youngest, which are the eggs, through to the larvae, the pupae, the emerging adults, and the adults. And these go on for genetic analysis. And this is just a photo to illustrate the diversity of the nests, well, some of the nests that we got in 2023. And you can see that some of the nests are quite small. They're perhaps the primary nests that were caught earlier in the season, or maybe they're nests that never really managed to get going. But some of the nests are considerably larger, For example, this one here in the middle is pretty beefy. The other thing you can notice is that a lot of them are associated with some sort of vegetation, so they have branches or twigs growing through them, and this just indicates where the nests are frequently found, embedded in shrubbery or in trees. But one thing I did find interesting is there's at least 2 which were found inside birdhouses. One of them in the concrete type birdhouse, and then another down here which seems to absolutely be pushing against the sides of the birdhouse. So the genetic methods. Back in 2016 we were first asked what genetic analysis we could do on the hornets which would help some of the management efforts. And so we took some microsatellite markers from the literature. So in France, they had already developed some microsatellite markers. There was this Arca et al. Paper from 2012. So they developed these markers that are neutral. They're not associated with any particular gene. They're not under any sort of selection. And they're distributed across the whole genome. And there's 15 of them, and their microsatellites are known for being highly variable. They're not actually highly variable in these Asian hornets because the population is so inbred and, well, it has got so little genetic diversity. But you can use these microsatellite markers, all 50 of them, to build up a genetic fingerprint for each of the hornets that we genotype. Because we've been using the same markers since 2016, we've got an extensive database that allows us to compare between years in the UK, and we're also kindly provided with some samples by Florence Mougel, who worked on, was a co-author on some of these French papers, and that's allowed us to compare our data directly to the French data. So we mentioned previously that the nest, we take samples across different life stages, and this just to illustrate that. So here we have an egg. I'm not sure if you can see, there's a red arrow pointing towards a small grey little egg at the bottom of the cell, which I'm sure is actually very hard to sample without popping. So we should be getting 10 eggs from each nest. We also get 10 of the larvae, these rather grub-like things here, and 10 of the pupae, which I always think looks slightly alien. And then we get 10 of the teneral or emerging adults, which are adults that have undergone pupation but have not yet escaped from their underneath the silken caps. And we also take 10 adult males and 10 adult females per nest. Obviously, we don't always get all of these life stages, but we try and get up to that theoretical maximum of 60 individuals per nest. So what do we do with this genetic data that we're collecting? One of the easiest things we can do is we can use the microsatellite genotypes to tell us whether the individual we're looking at has 1 copy of each chromosome or 2 copies of each chromosome. And we can do this very simply. If there's only 1 variation of that microsatellite for that individual, for all of those 15, it's highly likely it's only got 1 copy of each chromosome. But if we see any signs of heterozygosity, which is 2 different variants of that microsatellite, then we know there must be at least 2 copies of the chromosome present. Why is it important? It's important because the Asian hornet, like the honeybee, is a haploid-diploid system in which the individuals with 1 chromosome copy, the haploids, are the males, and the individuals with 2 chromosome copies, the diploids, are females. That is both workers and gynes and queens. And the way it works is inside the female, she makes eggs by a process which puts 1 copy of each of her chromosomes into the eggs. If she chooses to fertilise the egg with sperm from the male that she mated with, she passes on 1 copy of her chromosome, it combines with a copy of the chromosome from the sperm, and it forms diploid individuals, both of which will develop into being females. However, if she takes the egg and doesn't fertilise it, there's just 1 copy of the chromosome inside each egg, and that will develop. It's a haploid, and the growing gamete will recognise it only has 1 copy of each chromosome, and it will develop as a drone. There is a quirk in these haploid-diploid systems where in areas with very low genetic diversity or instances where there's a lot of inbreeding, some of the fertilised eggs think that they are haploid. The mechanism that they're using to determine whether they've got one copy of each chromosome or 2 copies of each chromosome is fooled into believing they've just got a single copy, and they develop into males called diploid males. And this can be quite problematic in some of the populations, which I'll discuss in one of the coming slides. We have used our genetic markers to identify the haploid individuals, and these are the individuals the Queen has laid with the intention that they will become drones. So we've taken the nest and it's been destroyed at a particular point in time, and we can count up how many haploids we have and how many diploids we have at each life stage. So in this example here, 4 out of the 10 eggs are haploid, 6 out of the 10 eggs are larvae, 8 out of the 10 eggs are pupae, but only 2 out of the 10 teneral adults are haploids. And there are a few adult males, I'm not sure if there are 10 or not. One of them, at least, is a true drone, a true haploid. And the reason we're interested in this is that if you think about the life cycle of the Asian hornet, so where you have the hibernating queen emerges, she forms a primary nest and builds up a supply of workers. They go on and they build a secondary nest. The population builds up, the nest reaches a large size, and at that point the queen switches from producing the workers, which are going out to provision the nest and make the nest larger, into producing the drones, into producing sons that she wants to go out and produce the next generation. After she's produced the drones, it's thought she then goes on to produce the gynes. So she'll swap from she's producing diploids here, they're going to be workers, haploids here, which are going to be the drones, and then she swaps back to producing diploids, and these are going to grow up as the gynes. And if we know what the nest is doing, whereabouts the production of the haploids are, we get a very good idea about whether the nest has been destroyed in time. So if you take this example here, there are some adult drones already produced, so that represents a certain risk of breeding, but it seems less likely that the gynes have reached adulthood, because it's likely that they're being produced at this point here. So there's a peak of production of these drones, and then she's swapping back to production of diploids, and it seems likely these, if they reached adulthood, would be the next generation of gynes, the next generation of queens. But the nest has been destroyed at this point, so none of these diploid individuals made it through to adulthood. And that's one of the main bits of information we get out of identifying haploids and diploids. It's about understanding whether the nest was destroyed before it started breeding. The other main use we make of the microsatellite data is we genotype every single lone hornet that we're sent, if we can. So everything is sent in ethanol. Sometimes they're from traps, and occasionally they're so degraded, they sort of smell pretty rank and they've started decomposing. Occasionally we can't get a genotype, but we work very hard to try and get a genotype from every single lone individual. So if you think about the work that we're doing, we produce up to 60 genotypes from each nest. So we have a thorough understanding of the genotypes that are present in the nest. We can infer what the queen's genotype was, so what the mother genotype was, and we can infer what the father or father's genotypes are. We also have the genotypes of these individual hornets that have been caught out in the wild. And what we can do is we can say, were those lone hornets from that nest, or were they from another nest? And we can do this very reliably using the mother and father genotype and just asking the question, can they be the offspring of these mothers and fathers? Or can they be the full siblings or half siblings to the individuals from the nest that we found and destroyed? So that's very handy. The key bit of information we're trying to get here really is we're trying to identify if there are any individuals out there that don't allocate back to a particular nest. And the reason these ones are important is that if we get this information in time, we can feed that back to the inspectors on the ground, and they can continue monitoring, or they can extend monitoring efforts. They can take some form of action to try and pick up on where these individuals have come from. But also, if it's not possible to find the nest that these individuals have come from, that's an identifier that there's potentially going to be further nests in subsequent years. Because if there is a local nest nearby that these have come from and it remains undiscovered, then potentially it will go on and breed. And this analysis gives lots of relevant information. So the results, or if we take the results firstly from evidence that nests were destroyed later on in the season and that they may have started breeding. This is the mix of the morphological data, which identifies the gynes, and the genetic data, which identifies the true adult drones. So these are the males that have reached adulthood and are haploids. So then none of them are these diploid males. And the figure here, we have the adult drones are just red spots, locations where we found the adult drones. And then the map is not fine enough resolution, so I've had to condense down. The sites where we found the gynes have got, in theory, the blue circle, but it's gynes and drones. It's a filled blue circle. So these places here are ones where we found both gynes and drones. We found 5 nests where there were gynes. There was one quite far to the north, and then some down in southeast England. And we had 9 nests which had true adult drones. This is slightly what you would expect. If the drones are being produced first, we're expecting that we will find more nests that had the adult drones than we would find the gynes which are being produced later. And in 4 of the nests, we found both adult gynes and adult drones. And just as an observation, when we found the gynes, they were mostly outside the nest. But when we found the drones, they were mostly within the nest. It's just an interesting observation. Moving on to the results from the lone hornets. With these lone hornets, we were trying to genetically allocate them back to a nest that had been destroyed or not. And we genotyped a total of 1,664 lone hornets, of which 1,627 we could allocate back to one of the nests that had been destroyed. And this left 37 hornets that we couldn't say which nest they had come from, and it seemed likely they were from a nest we hadn't sampled. 4 of them were spring caught queens, so they were caught in the early spring months, and they're likely to have been mated females, mated queens who were flying around trying to establish a nest. There was some monitoring nearer by these sites, but it's unlikely that there was an active nest. They had yet to form the nest themselves in 2023. 3 of the summer caught ones were singletons, which is to say they were hornets caught on their own, far from other hornets. 2 of them were drones, so they were the males out trying to find mates, and it's likely these had travelled some distance from their nests, or they could potentially have been hitchhikers. It's not very clear. And 1 of them was a worker. There was some monitoring around these sites, but no other sightings. Whilst these definitely have come from an active nest, whereabouts that nest was is quite diffuse. We don't really know where the nest was. There were no other hornets caught, so they could have been accidentally transported as hitchhikers or something. But at 6 locations, we had clusters of 2 to 14 hornets. So we were catching multiple individuals, likely from the same nest that we had not managed to find. And these were found at 5 locations in Kent. So again, that southeast cluster, and 1 location at Plymouth, where we found 3 adult individuals, which was a relatively strong indication that there was a nest somewhere that had yet to be discovered. Just combining those 2 bits of information into a single figure, into a single slide, we have both the unallocated hornets and which indicate where there might be nests missing, and we have the nests that have potentially produced the next generation of queens and drones. These results are all given to DEFRA Bee Health and to APHA Bee Health to inform the management decisions they make about what to do this season, so trapping and monitoring. In many ways, the results I've just presented are the bulk of the results that we're interested in. So the reason we undertake the genetic analysis and the reason why DEFRA pays for the genetic analysis is for the information that's relevant to the management of the Asian hornet. It's extremely fascinating to be working on Asian hornet genetics, but that in itself isn't a sufficient reason to do it. But 2023 had a large amount of data compared to previous years, and we were able to also look into some of the other things that were happening in our data. And one thing I was particularly interested to look at was whether what we think is true about the life cycle of the Asian hornet in its reproductive stage is actually true. So what is thought happens is that the female will start to produce the haploids, the drones, and then she swaps to producing the gynes. And so I was interested to have a look in our data to see whether this was true or not. So what we did was we made a graph that showed whether nests had haploids up here or no haploids. And this is a progression over time where 0 is the first nest we caught and then days since that first nest was caught. And our statistician fitted a regression to this to show at what point the haploids were being produced if you fitted it to a curve. So if you look at the haploids first, there is fairly early on the haploids are being produced. If you then plot the presence of true drones, again saying nests with no true drones, nests with true drones, and these have been checked to make sure that none of them are diploid males, the fitted line occurs later on than that for the haploids. And you would expect this to be true because the haploids are just true drones which haven't necessarily reached adulthood yet. And so they take a while to mature and then they turn into the true drones, the male haploids. We can also plot on the presence of gynes. Again, it's nests with no gynes, and then this is the nests with gynes, progression over time. And you can see that the fitted line occurs later on in the season than it does for the true drones. And so we do have this slight step in time, which is supporting this idea. It's supporting the idea that you get the production of the drones first, followed by the production of the gynes, as you can see from the emergence of these curves. And this is reassuring because if we had found evidence that gynes were actually being produced before the drones, then it's possible that some of the nests where we thought they were at a lower risk of breeding were actually quite high. But that doesn't seem to be the case, so that's reassuring. One thing that comes out from that is the presence of these diploid drones. The female queen, the queen, is laying these diploid eggs with the intention that they're going to develop into workers, and they don't. They develop into drones, and they're unlikely to be performing the function of a worker. They're less likely to perhaps go out and forage. They're perhaps less able to defend the nest, and they're also going to be another mouth to feed within the nest. So it's likely that represent a drain on the resource of the nest and a fitness cost. And they are present in quite a high proportion of the nests that we found. So in 2023, we found them in about 1/3, 28% of the nests, which compares to the numbers from Arca et al. In France at 33%. So again, around 1/3. So it's consistent across the two places. But it's not only that, but we also found nests where we found the presence of triploids. These are individuals with 3 copies of each chromosome. They're workers, and they seem to be no different to diploid workers. And they must have been produced when a female queen mated with a diploid drone, and that will give rise to these 3 chromosome copies. But if those females in the nest were laid as gynes, when they went on to reproduce, they wouldn't be able to lay viable eggs. So the cost of these drones is not only the cost to the first generation nest that they're in, but also they carry a cost into the next generation if they manage to breed successfully. I mean, the indication is that they aren't breeding successfully very often, but even so, that's a sort of second generation cost. It's been suggested that the queen, to get round this issue of the diploid drones, a mechanism could be to mate with many different drones. So the genetic cause of diploid drones is low genetic diversity. If the queen mates with a drone that has the same sex-determining locus allele as she has, that's when you get the production of the diploid drones. At least that's the hypothesis. So the idea is that if the queen mates with multiple males, she's more likely to produce at least some individuals that are proper workers and fewer of these diploid drones. But we don't see this in the UK data. Last year we had 91% of the nests were single mated, 9% of them were double mated, and there was no evidence at all of triple mated individuals or higher numbers of matings than that. And this is very similar to the results we found in previous years. We've never yet really been confident that we found a triple mated nest. We have had some double mated ones, but it seems like it's predominantly single mated. Now the numbers in Arca et al. In the published paper are a lot higher. They report a very high proportion of a higher number of multi-matings. They use a piece of software called Colony to estimate the number of multi-matings that you need to account for the data in each nest. And when we apply that same framework of analysis to our data, it also comes up with higher numbers. But if you look at the raw data, at least when you have the single and the double mated queens, it's very easy to see and easy to analyse that you actually only need to have, say, 1 or 2 drones accounting for that. So for that reason, we don't tend to use Colony to estimate the number of matings that's going on. If you had higher number of matings, so higher numbers of fathers contributing to the nest, it's not possible to manually determine the number of drones, and you definitely need to use this a program like Colony to make that estimation. If we reanalyze the Arca data using the same methods that we typically use, then the multi-mating rate does drop, and in fact around 44% of their nests could be accounted for by single matings, about 11% by double matings, and then around 44% are triple mated or more than that, females. So it's possible that in France, in the area that Arca et al. Studied, there is a higher rate of multi-mating than we find in the UK, but it's hard to say which is typical for the Asian hornet. It would be interesting to see further data across the rest of Europe to see which one occurs more frequently, the sort of low rate that's found in the UK or the slightly higher rate that's found in France back in 2015. So from our data, we don't see any sign of higher rates of multi-mating to recompense for this. Another thing that comes out in our data that's not related to the double matings is that we do see quite a large number of nests that have worker-laid eggs in them. About 13% of the nests had worker-laid eggs. The workers lay the eggs, they haven't mated, therefore the eggs have to develop as haploid and drones. But genetically they're quite easy to pick out, provided we have enough data in the nest. We haven't done any further analyses to see whether there's something about perhaps the nest is smaller or it's failing or it's later in the season to try and account for this, but it's certainly an interesting observation. So to just pick up the strand of these diploid drones a little further. So we know that diploid drones are present in about 1/3 of the nests, and they're likely to be present when the level of inbreeding is high. And there are some papers that talk about the potential for what's called a diploid male vortex. It's a sort of extinction vortex, which takes place if the population has too few sex-determining loci. You get too many diploid drones, the diversity of the population decreases and it enters an extinction spiral and it's unable to breed any further. So it's possible that some of these very remote nests, where they do seem to be singletons in the landscape, they might die out anyway, because if the gynes are forced to mate with their brothers because there are no other drones nearby, they're more likely to produce these diploid drones and the nests might simply not be viable. And that might explain why you seem to get sort of jumps in the presence of the Asian hornet in continental Europe. And then they they seem to disappear, and it's only as the invasion front gets closer and there are more Asian hornets dispersing into the location that you get established population. But you certainly wouldn't want to take it as read. You certainly wouldn't want to be bold and say, well, these will die out anyway. They definitely need management effort, and management effort is fully expended in making sure they do die out. Final thing that we check every year is to see whether the hornets that we find in the UK are likely from the European population, or whether it's possible that we're also looking at recent arrivals from Southeast Asia. And so far we found no evidence at all that we have got any Asian hornets coming in from Asia rather than the European population. So our current activities partly is just tidying up the loose ends from the previous season in terms of getting all our reports completely tidied away, but also preparing for the next wave of Asian hornets. So I think we've already received our first spring court female, but we're likely to get quite a few spring court females this year. And one of the central questions we'll be asking with those is, are they the offspring of any of the nests that we found and destroyed in 2023? So we've already highlighted places where we think the nests were destroyed too late. So it'll be interesting to see whether we find spring court females from those that we can tie back from those nests. And that's a very straightforward question to answer with the genetic markers we've got. We're very well able to identify things back to the nest that they came from. We're also getting prepared for the large volume of samples we're expecting to receive in summer and into autumn, and possibly even into early winter. So the number of samples we got last year was huge compared to the number of samples that we got previously. And so we're just making sure that we're going to try and streamline everything, make everything work more efficiently, and hope to get the data back quicker for the genetic results. We also have a PhD student who's working on an entirely different track, is working on a different set of genetic markers. So we were funded by DEFRA to do the whole genomes of some 10 Asian hornets and to use those to see if we couldn't develop better or different genetic markers. In the project that we did, we found genetic markers a low number, and they performed the same as the microsatellites. We had a plan to move across to those, but thankfully we didn't, because we'd have found it a struggle to deliver both in 2023. But we have a PhD student who is developing a panel of markers, some sort of 400 or 500 markers across the whole genome of the Asian hornets. And the hope is that she'll be able to look into questions like about kinship in far greater detail, and perhaps see if it's possible to get some sort of better resolution about where the Asian hornets are coming from. So thank you very much for listening to the talk. And I'd just like to acknowledge all of the people who contributed to the work. So beekeepers and members of the public for their vigilance and for their reporting, without whom it would have been extremely difficult to find Asian hornets. To DEFRA for the funding, and the National Bee Unit, particularly the bee health inspectors, for going out and finding these samples, and for returning all of the samples in good condition and in a timely way for Fera to be able to do the analysis on it. The work took in a huge amount of Fera staff this year because of the sheer volume of samples we were dealing with. Some 4,600 samples were genotyped. So at the National Reference Laboratory, particularly Victoria Tomkes and Ben Jones, and from entomology, Rob Deedy has been leading up much of the work on these Asian hornets. And we actually had to call across two molecular teams, the R&D team and the high throughput delivery team. And just to name check, Sam McGregor and Joy Kay from those teams as well. But we also had to use the land use team for mapping and figuring out where everything was from. And thank you very much for the BBKA for giving me the opportunity to talk today. Unfortunately, I can't take your questions. I have been sent 4 questions, which I'll try and give you a brief answer for now. But I think you'll be joined by some colleagues from DEFRA after this session who might be able to answer some of the questions or forward them on to me. A common question is there a resident UK population? And from the genetic data, we cannot confirm that there is, and nor can we say that there's not. And there's a variety of reasons for that. One of them is that we don't have good comparative data to compare what we might call the UK population against. So if we want to say there is a UK population and it is different from a population in northern France, or it's different from the population in Holland, then we need to have good microsatellite data from those locations. The papers out there, perhaps the most relevant one is the Arca et al. Paper from 2015, but that's 9 nests taken in 2015 or before from, I think it was more central France. That doesn't represent a good reference data set for us to compare against and say it is different or it's not different from the UK population. So that absence of a reference population means that we can't make that comparison very easily. The other problem is that we're using 15 microsatellite markers in a highly low genetic diversity population. And so we could look at questions like kinship of the queens from nearby nests. So if we took 2 nests and we said, are the queens from those nests full siblings to each other, that might help us say whether there was a nest that had been breeding in the previous year. But unfortunately, with our current microsatellite markers, we can't tell the difference between 2 random individuals that are not related to each other and 2 siblings if we're only looking at those 2 individuals. So unfortunately, the data cannot tell us whether we have, and also cannot tell us if we haven't got a resident UK population. Could a single mated or inbred queen suggest we have a small population in the UK which are breeding? Has the analysis been cross-referenced with the results from France to confirm genetics? So yes, we do make reference to that French paper Arca et al. From 2015, and we've also run samples from that so that we've got directly comparable data sets. But their data set is quite small, and it's also from quite a while ago. So I discussed in the previous slide, so it does appear that we have fewer matings compared to that Arca et al. Paper, but we can't tell what the norm is. Is it that Arca et al. Were picking up abnormally high rates of mating in the population that they were sampling, and the UK is more typical of the inverted commas normal situation, or is the UK the outlier? So it'd be very nice to see comparative data sets from the rest of Europe to answer which one is perhaps the oddity, or whether it's a bit of a continuum and we've perhaps got a sampling bias in one of the papers or one of the studies. I don't really see that anyone's going to do that study, but it would be very interesting if it did happen. I don't think that we would be able to use that information as it stands, the information just what we've got from our current study and the Arca et al. Paper, to say that there is a meaningful difference and that one is suggestive of a resident UK population. You suggest the queens have different mothers, suggesting we don't have a population here. Has the gene analysis been compared with the results from France to confirm this? Inbred single-mated queens suggest that these could be from a small population in the UK. Well, the second part of the question is very similar to the question above. We can look at the genetic diversity index for the population we have, which is a sort of inbreeding index, and we could compare it to the Arca one, but we are comparing 2 quite small data sets to each other, and it's not hard to know if that comparison will be meaningful. And the same with the single mating versus double mating versus multi-mating rate. Again, it's hard to say that that's a meaningful comparison that means something significantly different is occurring. For the first part of the question, you suggest the queens have different mothers, suggesting we don't have a population here. So in some locations, for example in Maidstone, we had many nests, sort of 5 or 6 nests in a single location. And what we did was we asked the question whether the mothers of those nests could all be full siblings to each other. And when we're comparing more than 2 individuals, we begin to be able to say with greater confidence, these cannot be full siblings. And that was the case in many of the sites where we had lots of nests in close proximity. They could not all have come from a single nest in 2022. They could have come from a nest in the UK in 2022 with genetic input from elsewhere, so perhaps drones from outside, or they could all be freshly imported individuals. But the genetics can't really inform on those different scenarios. All we can say for sure is that the queens that formed the nest that we found in 2023 in that small geographic area didn't come from just one nest in 2022. Another group of questions that I get is, can we use different methods? Would approaches like whole genome sequencing help study the Asian hornets? And the answer is possibly. Whole genome sequencing is not practical when we talk about the number of samples that we would propose to do for Asian hornets, taking into consideration how large the genome size is. So it'd be extremely expensive to do. It'd be problematic to store all that data. And I'm not sure that we would actually be able to, we have the analytical framework to assemble those genomes and then compare them to each other. But as I've said before, we have a PhD student who's looking at genomic type approaches. She's looking at large scale SNP markers across the genome. And it's possible that those markers might be able to say something informative about whether if you took 2 queens, you could say for sure that they're full siblings or not. And so it's possible that other approaches might be able to solve it. And the PhD student with Newcastle University might be able to give some answers. I think I'd best end there to leave time for questions from the DEFRA representatives. Thank you very much.
Thank you for that excellent and very interesting look at the genetics of Asian hornets. Now we have Professor Nicola Spence and Dr. Belinda Phillipson on Zoom with us.
How can we confidently say they haven't come from Asia, but we can confidently say they've come from France?
Because the genetics, we know that they're very closely related to the population that are derived on the continent, and that's quite strikingly different. The genetics that you've seen from the ones in Asia, with the differences are bigger. So
why can't we say confidently whether we've got a population here or they're coming in from France?
Because they're much closer. It's really tricky, but they're much more closely related. So there's a bigger distance. If you think in terms of time of when the first one arrived in France and then spread, that one's just spread and generated the population here. And the differences between the ones that we see in France and the ones we see in the UK are really, really small. So it's really, and populations are normally done as a distinct population that's split off of an initial one and then separated as a population over a period of years. So it's an ongoing wave, if you imagine that it's spread across continental Europe, and now we've got incursions coming into GB.
Is it really valid to talk about an extinction vortex when France was started with 1 queen?
But she was multi-mated.
And the queen that's arrived here is also multi-mated?
No, we don't think we've got data that demonstrates that the queen has mated with any more than 2. The one that arrived in France was initially multi-mated with 5 or 6, is my understanding.
Does that help? Can we put any restrictions on imports and movements to prevent further ingress to hopefully produce an extinction vortex?
Asian hornet is a really good hitchhiker, and that's what the results from the initial risk assessment showed. So even if we put a ban on imports, they could come in. You imagine the number of people who go on camping trips in France. They can hide in the arches of cars. We found them in camping equipment. It's just impossible to prevent them coming in.
So extinction vortex is probably unlikely because we'll just be having fresh imports. Anyone else? Does it matter if they are double mated, single mated or triple mated, if whatever mating they are, they're going to decimate our insect population?
In the long term, the more genetic diversity they have, the better chance they have of spreading further, in a sense, creating more impact. If it's in a biological sense, it's quite an interesting population, because you could argue that with just one queen that arrived in France in the first place, it would end up in being a bit of a population dead end, and it wouldn't survive. But clearly that isn't the case. So it's obviously very capable and survive and can spread and survive. So I agree in the sense that we shouldn't worry so much about the multi-mating, but it tells us a bit about the edge of the population and how likely it is to survive. And it's additional information that we can pick up when we destroy the nests and analyse them.
Well, was the one from France well mated or poorly mated compared to where they come from?
That's a good question, and I don't know. I'd need to take that away.
Right. Well, that'd be a good question for you to come back on if there's research on
Indeed. That I would need to take to Ellie and the other experts.
Do you collaborate much with the universities in Europe, particularly where Sandra Ross's Noriel comes from?
We do, but we also liaise. So there are researchers in the UK. So Pete Kennedy at the University of Exeter works closely with Sandra, and it's a small world in terms of biology and research. So we, even if we don't speak to them regularly, certainly our UK researchers who we have good connections with do.
Why is the government not promoting it as an environmental and economic issue rather than the beekeeper issue?
I don't think we are promoting it as a beekeeper issue. It's clear, we've said in all of our messaging that it not only has an impact on honeybees, it will also have an impact on unmanaged and wild pollinators.
As part of the government's environmental improvement plan, this is one of the high priority reasons why we're protecting pollinators generally from Asian hornet. But obviously in this instance, there is a specific risk to honeybees, but we know that the wild bee population is also at risk.
There's a feeling that's not coming across to the general public.
That's tricky because of some of the messaging is not within our control. The media in particular are very fond of honeybees, as we all are, and they are like a social insect species. So I think they think that will raise people's concerns much more highly if they think that honeybees are under pressure or under attack.
But I think it's a good point for us to consider how we better communicate the risk to the wider environment and the wider pollinator community. So thank you for that point.
And I think the economic indicators as well are at risk. There's quite a lot of factors there. I'll come back to Chloe, but over there. Is the Asian hornet on the National Risk Register?
It isn't, no, but we have flagged it up in various risk places, and there are local resilience forums which we've been flagging up the risk to. There are quite specific criteria that you have to go through in order to get something on the National Risk Register.
Will you be updating the National Pollinator Strategy that expires this year with more focus on the invasive species that threaten the native pollinators?
The National Pollinator Strategy is not something that sits, it's not my own responsibility. I do contribute to it, but it's not my own responsibility. So I'm sure that we will be including that in the list of things that will be part of the new and updated strategy.
Do we know anything about the diet of Asian hornet, the balance of it across the seasons, as there's been some DNA analysis?
There's been a small amount of DNA analysis, but it's not been huge. So we know about some of the species that they like to eat, but I don't think it's been done across the season. It will have just been done with a snapshot in time of Asian hornets that have been captured.
think if no one else has got a question, I'm going to come to Lou.
Hello, Belinda. My question is, we know that we have gynns overwintering in Kent, as one has already been found in a potting shed. How many do we think potentially will overwinter successfully and go on to make primary nests this year?
You mean how many primary nests we would expect to see this year?
Yes, from what was missed last year.
I think that's incredibly difficult to say, and I would really like to say that we wouldn't expect to see any, because the National Bee Unit and APHA did an amazing job in destroying 72 nests last year. I'd need to double check the analysis of the gyn that was found in the potting shed to see whether they've managed to allocate her to any of the nests this year. And that answer I'd need to look into and get back to you. But
So,
I don't think we can definitively say that she was produced from one of the nests that was found or destroyed in GB last year.
So what are the chances that a nest went totally off the radar and didn't get discovered last year that has produced gynns?
I think that's really hard to say. That it's a diminishing law of return. So in terms of a nest produces a large amount of gynns, and then a smaller number of those are fertilized, and then a smaller number of those overwinter, and then a smaller number of those go on to produce successful nests. So it could be that there is the odd one or two nests which has been produced in GB but has not been successful in making it fully through to a developed nest that then has gone on to produce more gynns. The chance, it's really difficult to estimate.
I mean, that's part of the reason why we're doing early spring trapping this year, to satisfy ourselves that we don't have any of these populations that may have overwintered, and to pick up any that have.
Brilliant. One last question. I've spoke with Alistair Christie in Jersey quite extensively over the last couple of years, and his numbers seem to indicate that between 10 and 15% of gynns will overwinter successfully. Can you tell me how many gynns have been found in nests that have come into you guys that have been investigated?
Well, I think I don't know. I don't have the total number of gynns, but I think Ellie presented it in her slides. So there was 4, 5 of the nests had gynns.
And do you know how many they had in them? Sorry?
I off the top of my head, no, but the lab will have that information.
Were they in the hundreds?
No.
Okay. Okay, thank you very much.
Are there any extra questions from anyone? No, I think that's it. Thanks, Belinda and Nicola, for answering questions.
Thank you. Okay, thank you. I hope the rest of the presentation goes well. Bye-bye.